Happy World Fragile X Day to everyone. I want to thank you all for joining us today to commemorate this very special occasion. Peter, this is really a remarkable time to be a neuroscience researcher, especially in the field of developmental disorders, rare diseases, and particularly Fragile X. You've led the way in studying and comparing different single gene causes of autism. What has you most excited right now?
Well, I think over the last 10 years, we've really defined the genetic landscape that leads to at least the monogenic forms of the neurodevelopmental disorders. And we now know that they cluster around sort of subcategories of gene function. And we know there's anywhere between 200 and 1,000 genes that can lead to neurodevelopmental disorders like autism and intellectual disability. And that arms researchers with the tools to make animal models. And genetics has also gone a long way that we can make any model we want to now. We have human stem cell models, we have rodent models, we have large animal models, and we can really address questions that were never achievable before. If you combine that with the new techniques for being able to understand brain circuits, where we can delete a gene in a particular cell type or in a particular cell region, we can really now start to define the pathophysiology associated with these disorders. And armed with that, we can ask much more fundamental questions about what's going awry in the brain in the absence of a particular gene. So it's a really exciting time to properly define these disorders in terms of how they affect the brain, and also ask questions of when can we ameliorate the pathophysiology. So it's a phenomenal time to be a neuroscientist working in this field.
Liz, this seems to be a very busy time for clinical trials in Fragile X.
Yeah, it's very busy. We have quite a few different industry partners that are looking at different targets. And so we're working hard to figure out what are the best ways to go. And I think in the Fragile X world, we've had some failed trials in the past because we were really the first condition that went toward trying to target treatment of the underlying disorder and sort of fix the brain and fix the actual condition itself rather than just using supportive treatments. And so we learned a lot during those trials about problems that we had with the models that were being used at that time, really 10, 13 years ago even. We made a lot of progress on new ways of approaching these trials that hopefully will allow some of the agents that are going forward right now to be more successful.
Well, things certainly have progressed since the early days of Fragile X clinical trials, and I think you deserve a lot of the credit for that, Liz. What would you say is the state of the art for clinical trials methodology in Fragile X? And just to extend that, what can other rare diseases learn from this progress?
Yeah, so I think we've learned that you can't just, take any outcome measure from autism and use it in Fragile X. And we've learned a little bit about what patient populations we can use. And I think one of the things we've learned is that the mouse model in particular is very easy to treat, and we can see improvements in the mouse model easily, but then that's very hard to translate to people due to the variability of the condition in people and the heterogeneity of their background and the heterogeneity of where they live and what they do. And so the variability makes it challenging to use measures that, have a lot of variability themselves. And so we've done a lot of work to find objective measures that are going to look at the Fragile X condition better. Things like cognitive measures, like the NIH toolbox that we've now spent about 8 years validating for intellectual disability and particularly for Fragile X. Things like EEG that we can measure, circuit activity or brain responses to sound, for instance, that maybe those are not kinds of things that we can get a drug approved on But they are the kinds of things that can give us an indication that we have a signal, so we know which drugs to move forward. And we don't know that circuits and different neurotransmitter mechanisms and so forth are going to behave exactly the same in the mouse and the human. But if we have ways of moving forward into humans where we do, where we have some sort of directly translatable markers, like an EEG finding that's the same in the mouse and the human, and we can show that a drug targets this exactly the same way in a mouse and a human, then that makes that a little more translatable to humans than some of the other mouse behaviors we've been relying on. And also, we are now thinking that, you know, we get data in a model organism, and that's great. We really want to have that data to be able to show that a drug looks promising, but we've changed our thinking that you don't just go into a big trial. You need to do an early phase trial where we look at a lot of objective measures and demonstrate that we can see a signal. And if we can't see a signal in a trial where we've looked at what the drug does in a smaller number of patients with objective measures that have less placebo effect, then we may not want to use patient resources to do a large trial of that agent. And I think this strategy of doing a small trial with kind of intensive use of different measures to prove that there's a signal has actually been fairly successful over the recent years. And I think we'll see more successes with that as we go forward.
It is really fascinating to think that you can use human electrophysiology to actually get a glimpse of what's going on in the brain. That seems like a really powerful tool. What are the specific things that you're looking at when you look at human electrophysiology?
So we're looking at both at EEG and something called ERP, which is event-related potentials. And so in Fragile X, we look at event-related potentials mainly related to sound. And that's because patients with Fragile X have an extreme sensitivity to sound in many cases. And when you actually measure their response to sound in the brain, they have a bigger response than you would see in a control person of the same age. And so we can measure this excessive response to sound. There are a lot of characteristics about sound processing surrounding that we can measure. And then there's an EEG background. There's something called gamma power, which is just a certain frequency of EEG activity that is increased in Fragile X. And so we've been able to see in the mouse model that the gamma power is abnormal, just like in the people. And we've been able to see in the mouse model when we use really good detailed EEG that uses multiple leads and not just, you know, a couple leads on the mouse head. When we really look at the mouse EEG, we can see the same kinds of abnormal processing of sound in the mouse model as well. So if we have an agent that changes those things in the mouse, then we can expect, and then we can immediately go and check and see, does that agent change the same thing in a person? And then what we really need to do, so this is work that we don't quite have done yet. What we really need to do is be able to hook up those EEG changes to clinical phenotypes and show that, like, when you change gamma power, you get less anxiety or something like that. Or when you change novel sound processing, you get better language. But we aren't there yet, and there's ongoing work in that direction that hopefully will help us in the future with seeing these EEG changes and knowing then what should we measure clinically that will be clinically meaningful.
So there's still a need to validate some of these electrophysiological methods and to see if they correlate with clinical improvement. But is there a sense that some of these electrophysiologic tests will actually normalize with treatment? Have we seen responses to drug treatment that actually involve changes in electrophysiology?
Yeah, I mean, there are several examples of that. Craig Erickson is running an NIH-funded project where they're looking at changes after a single dose of different agents, and they have seen that some of the agents that they have used have changed the electrophysiology. And we ran a trial of a phosphodiesterase inhibitor, a PDE4D inhibitor, recently a phase 2 study, which actually showed that you could change the excessive sound sensitivity in the patients with the drug. And so that's that's a very strong finding, particularly when in that study it was coupled with a change in cognitive performance on the NIH toolbox, which is another objective performance-based measure, and the parents reported that the patients were better. So this was a small, intensive, let's look and see if there's a signal kind of study, and this is an example of the kind of study that we need to do. So now we know we have a great early signal, and so hopefully that will predict better results in phase 3 trials than what we've had in the past.
That really does seem clinically relevant, and it really does seem relevant to quality of life as well. So that's very exciting.
Yeah.
Peter, one of the questions that I hear people ask most often is how treatable developmental disorders in general might be later in life, and specifically how treatable Fragile X might be at later stages of life. Now, you've been a leader in this field in studying development in Fragile X and a lot of other monogenic causes of autism. What's your take on this? How reversible is this later in life?
Well, that's the $6 million question. I mean, Adrian Bird really changed the landscape and the work of Huda Zogby as well, showing that Rett syndrome, when MECP2, the gene that's altered in Rett syndrome, is re-expressed late in life or knocked down late in life, you can either, in the first case, you can rescue many of the phenotypes associated with in the rodent models. And if you knock it out late in life, you can recapitulate many of the features of Rett syndrome, which argued that there isn't a critical period for MECP2 re-expression or Rett syndrome. Now, whether that's true for numerous other disorders, needs to be tested, and it is being tested for many disorders, but it's one of the central questions we're asking in my lab and in the center that I work in, and other labs around the world are asking. But I think the most likely outcome of all of this, our prediction, if you will, is that certain models will be more or less amenable to gene re-expression late in life, and we don't know. And the key question there, I think, as well, is it's unlikely to rescue everything when you express late in life. There's different critical periods. There isn't a single critical period. So certain features might be more amenable to genetic re-expression. Other features might be less amenable to that, and we won't know until we try. The other key aspect to that, I think, Mike, is that these are largely being tested with genetic re-expression. But even if genetic re-expression has a critical period and you can't rescue certain features of Fragile X syndrome, for example, that does not mean they're not amenable to other types of therapies. Pharmaceutical therapies might work on the circuit, which is still amenable to treatment, even though changing the root cause, i.e. the loss of FMRP, is no longer amenable to just re-expressing it. So I think there's lots of different critical periods. Different features will have different critical periods, different treatments will have different critical periods, and that's really important, and that's why we need just a lot more research into all of these different monogenic forms, including Fragile X.
Huge advantage for rare diseases, for monogenic diseases, for a lot of the things that you're studying, the single gene causes of autism. And this is a huge advantage when it comes to drug discovery and development of new therapeutics. It really is amazingly powerful.
It is. And we talk about the rare diseases, but if you combine all the different rare diseases together, you end up with a disorder state that is not that rare. It affects well over 10% of the population. So it's crucial that we understand these, and especially how similar they are, and if we can cluster them together for therapeutic purposes.
So what research still needs to be done to understand development and its role in these neurodevelopmental disorders?
Yeah, so what we're trying to do is cluster the different monogenic forms of autism and intellectual disability, not just in terms of commonalities in the biochemical pathways that might be affected, but commonalities of timing of when the brain circuits start to change, and to really identify, to cluster them based on developmental windows. And one of the things we found recently was if we treat early in life, just transiently, we can rescue some of the issues in our models of Fragile X syndrome, that we can remove the treatment and we've rescued that particular stage of development. And other people are starting to find the same things for other aspects of Fragile X. So I think that's actually a really key question that's never been thoroughly addressed, is are there windows that if we can treat during those windows, we can get good permanent recovery? And I think that's a really interesting question, again, about critical periods that we really need to understand more deeply.
Very exciting. Liz, I guess I should ask you the same question about what work remains to be done in the clinical trials field, because we've come a long way, but we're not totally done yet, I assume.
Yeah, we have a great deal of work to do in the clinical trials field. I mean, we always lag by, 10 years or so, the basic science people, in figuring out how to translate things to people, because it's just so challenging. But I would say that the work that needs to be done is, I think Peter's idea of critical windows is very important. And we in clinic have always thought that combinations of medications would be needed to really fully fix Fragile X, because the FMRP has so many roles and it may have different roles at different times in life. And there may be certain things that work when you're young and certain things that work better when you're older, as Peter was mentioning. And there may be things that work together. But in order to have that kind of treatment, we have to get our foot in the door. The reason cancer has so many good treatments and why they can add drugs onto other drugs is that they got drugs approved at the beginning, and you can only test combinations in general after something is approved, although we are doing some new study designs to try to test combinations of medications. And I think it's important to develop that. So that's an area of clinical science that we need to develop. But traditionally, you have to get approvals of one thing, and then you can treat a patient with an additional medication that's investigational. So trial designs for combinations, we've done a lot of work on outcome measures, but at this point, we don't yet have the FDA considering them, quote, fit for purpose. And this is work we need to do with the FDA so that we can generate outcome measures that index the phenotypes in Fragile X and index the important things to families in Fragile X well, in addition to giving us good objective performance-based measures in order to move drugs forward. So we have a lot of validation yet to do, even on the things that we've already done work on. And so I really see us as needing to continue to improve our clinical trial science. We've collected in the recent years a fair amount of well-vetted natural history where we can actually see the trajectory of Fragile X, of typical Fragile X with just standard of care treatment. What do patients with Fragile X do over time in terms of their development based on different measures? And we need to have more of that, because if we had a gene therapy, we certainly wouldn't expect all the effects to occur in the context of like a 3-month trial. We'd need to be able to look and see, what did that gene therapy do across life? And to be able to understand that, we have to have these very good natural history studies where we understand what does Fragile X actually look at across life on measures that we can use to demonstrate, what is the gene therapy or what is another drug doing. And so that's early in its collection. We have a new project with the CDC where we're going to collect 600 data on 600 patients that will be across the pediatric age span, really from about 5 to 23, which is going to give us very nice data on how does Fragile X evolve. We've already collected some data through my NeuroNext trial and through an earlier CDC project. And so I think we have a lot of building blocks to get to the place where we have this multiple therapy treatment that we really need. And right now we're in the midst of building the building, so to speak. We're just putting more bricks in the wall every year as we get that basic database that feels like cancer already have, that we can use to really get treatments approved and to improve people's lives.
It does seem like the validation of these clinical endpoints is a real catch-22. It's really even kind of a whole series of catch-22s because you have to correlate one thing with another thing, which correlates with quality of life, but then you have to show that it moves with treatment. But for it to move with treatment, you have to have a treatment that works, and then you have to just repeat this cycle over and over again. So where are we in that whole process? It feels like we're well on the path, but we're still on the path. We're not there yet.
Yeah, we're on the path. It's a challenge because yes, we have to show that we can do the measure in Fragile X and that it's reproducible and that the measures have content validity and the standard things you would do to validate a test. But then we have to show how much change on the measure is clinically meaningful. And to do that, we have to use interviews of families and other kinds of measures where families rate how much improvement they're seeing in the patient and whether that's a clinically meaningful improvement. So we are actually with NIH Toolbox and some of our family-based report measures of things like the biggest problem that they like, we have them just pick their biggest problem and then rate it on a scale. We're in active discussion with FDA on what we need to do to get those measures fit for purpose. And in the upcoming trials, there will be a lot of work toward providing FDA what they need to make these measures be what we need to get drugs approved. So it's an ongoing effort, and it really involves an interplay between us doing scientific validation on a measure and then going back to the families and asking them, well, if this measure changes this much and it corresponds to sort of this change in what your child can do you consider that a clinically meaningful change? And so the FDA is very focused on that right now, and that's work that is actively going on in the upcoming trials. So we're going to have each sort of 2 or 3 year period, we get more information on how to best move these forward. But yes, it's a challenge, but we're hitting it head on.
Well, here's a question for both of you, each of you. I want you to look into your crystal balls and to tell me where you see us in 5 years or in 10 years. Peter, where are we heading?
Yeah, well, I mean, I think for all the reasons I said earlier on, I think the future's bright. And from a fundamental research point of view, I think we're getting far better with our models of these disorders in order to get real predictive validity. I mean, that's the holy grail. If we can see a particular therapy having a beneficial effect in our animal models, that will be predictive of what's happening, what happens in the clinic. We have to get a lot better at the research level in making our models predictive. We need to understand our models better. We need to ask the model. We need to use our models in the appropriate way, and I think we're getting far better at that as scientists. So from that point of view, I'm very hopeful. I think we are going to have good predictive validity. We need a drug that we know or a treatment that we know works well in humans to test predictive validity in our animal models. And I'm really excited about the PDE4, that this might give us that opportunity. But yeah, with all of the new techniques we have, with all of the new knowledge we have, I think the future is very bright for coming up with therapies that are predictive in our animal models for the clinic.
And Liz, what is it that excites you about the future 5 years or 10 years?
Yeah, so I think we're turning a corner, and we know now that there are genetically based therapies in development for Fragile X, as well as small molecules. And it may be that these strategies will actually work best together, or different things will work better at different ages. And so we need to proceed ahead with all of that. And so in 5 to 10 years, I mean, I think we're just getting trial designs really under our belt. And I would hope that in that period of time, we have that initial drug approved so we can start adding things on. We're in the midst of doing gene therapy trials. We've got a process figured out for sort of fast fail, where we can look at animal data, move that into humans, and kind of a standardized approach to do a very, a small focused trial, figure out, does the drug work, and move on to a new target if it doesn't really seem to have an impact in humans. So and that's the way we're going to get to those combination therapies that we want. And so I think we have a lot of positivity going right now. I think we're moving into some big, exciting trials, and hopefully those will be more successful than the past. And I would hope that in 5 to 10 years, we've got at least one drug approved, and we're working on getting those add-on therapies, and we're moving actual genetic therapies like gene therapy into the clinic.
Well, it is a really exciting time, and it's a really hopeful time. And there really is this sense that everything is coalescing, that finally all the pieces are coming together. And I just really want to thank both of you for being here, and happy Fragile X Day.
Yeah, happy Fragile X Day.
Happy Fragile X Day to everyone.